Hi, so I'm from AbbVie. You may notice over on the bottom here, Business Technology Solutions. So I'm in IT, actually, not in the bioinformatics group or therapeutic area. We're actually in the data solutions area, so we're providing solutions to biologists, chemists, whatever, dealing with data and helping them move forward with their research. So Matt just asked me to talk a little bit about how we've used OmicSoft to build a sort of an omics hub. Historical omic data flow, microarray data, that would go into Partech. We had Rosetta Resolver. Proteomics data, we've got some max quant data that went into an Excel file and may just stay there. NGS data, that was going to our bioinformaticians. They'd had a local compute cluster. Chemical biology data, it's going to go into a BioELN at some level, Excel. The thing is how to do a cross-platform analysis or searching. There was no real good way to deal with, we'd have researchers doing something very, very clever, and they'd have some microarray or NGS results for their cells that were maybe acquired resistance, and then maybe they do some proteomics, maybe do a pull down, and then they would just sort through random Excel files or whatever. There's no good way to do a cross-platform analysis. And then further, maybe they do something that's very interesting. The project moves on. A couple of years later, they would come back and do a further analysis to dig in. With now they have more insight. Well, they would have to reassemble all the metadata with the raw data files again, and that wasn't kept together, right? How do you do that? And then further, let's say you really want to do these things and be clever about it. What's the access control? Maybe you don't have access to the cluster to get to the NGS files, or maybe all the Excel files are stored in your department share drive, but maybe the other person's in a different department. I can't get to that file. How do we do that? And so we were thinking, and by the way, that previous historical flow, that's where we were in 2014, right? So right at the end of 2014, beginning of 2015 is when we first started with Array Studio. So unlike Sean or JC, we've not been a long-time customer. We're thinking, what's an ideal omic data warehouse? What do we want to do? We want to be able to have a centralized, secure storage for data from all these platforms, right? Not just randomly here and there. And we'd like to have a sample and sample set database linked to those raw files. So we could say in the future, hey, do we have any samples on this kind of cell line, whatever? We'd like to have that in a database that we could access that in the future. And to make that work, we need to have a controlled, common vocabulary that's going to allow this cross-platform database to work, right? Know how people misspell, or they have short nicknames for the cell lines they use, or just the common typos. You need to have a common control vocabulary for all of these things. And then you need a flexible, centralized access control. You really can't be in the situation of each individual researcher has to contact 5 different people to get permission of, can I have your access? Can I access to your data? Oh, can I have your data? We needed a simple, centralized place. So the question is how to rapidly develop this. And so what I'm going to talk to you about is how we've approached that and how we've used Array Studio to try to get there. So building this AbbVie Omics Hub, we started with Array Studio on the cloud. Now that was an easy choice for us because at the time we'd spun up Array Studio as a demo with Matt on some little server we had. But to really run it, to have room for the lands that we for sure would want, we went directly to the cloud because we didn't have a local compute server to put it on. So just conveniently, that's when the cloud came out, and that's where we put it. With that, then we've got secure storage on AWS, where we can just expand and put all the various data types there on S3. And Much of the data is coming in from CROs. That's very easy. Rather than shipping us an encrypted hard drive, we just instruct them on how to set up a bucket. They share the bucket with us, and we get the data. And we share it all there. Now then, we also developed a controlled vocabulary for these analyses. Unfortunately, we didn't use exactly the same ontologies that the Curation tool is using with RayStudio, but similar. And we had our own reasons to choose DOID or mesh because maybe we had some samples in a biobank that was already developing and starting to use those. So ideally, in the future, we'll actually get data samples from that biobank, and we'll already have the terms work together. But then the key here is then to fully leverage the sample and sample set registration, right? Because then that ties all those things together. In a secure storage, they're available for RayStudio. Another thing that was important is then to bring in custom annotations to be able to handle all the different platforms of the proteomics data or whatever. And then further, there's always going to be people that want to do analysis in a different way that's just not natively part of RayStudio, or it's just their favorite way. We can still bring in those data tables or those inference tables into RayStudio. We still have the samples where the sample raw data files where they are. We know what the metadata is. They analyzed it. However, as long as they annotate that, bring the tables in, it's still all together. That's the goal. That's what we were working toward. Oh yeah, and then of course, if we do get a method that they've worked out that they really like, we've brought in R packages that can just live natively. And so we'll talk about Sention. We initially were doing that outside. Now it's a button. So that's been our approach. And we have the flexible access control. We don't have the LDAP yet, but we certainly at least actively use the controls for every view for access to the data files in the sample in the file browser, and also to the lands, and we use those to control access. So one of the things we developed was an RayStudio data in tool. It may look a little bit like the Curation tool because we basically took some of those same fields and built a tool around it using control vocabularies and also just a customized list of terms of things that we use in the organization. And this web tool, of course, it's built on the control vocabulary in a database. So we have in-cell term lookup for some controlled terms that we use, various study aim, for example. We also have validation. So you can see here somebody typed in, you type in M for uppercase male, not lowercase. How do you get the cell line properly labeled so we can map that? So it took a while to work out how to do this on the web, but in this case, Aaron Hickey worked this out. So we have this tool to facilitate, to facilitate this RayStudio data in. Now, at the end of this process, we still end up with a text file, sample set, samples. Currently, it's a manual register the samples, but then the very next step would be to just, if they're satisfied, send it directly for sample registration. We don't have that yet because, of course, it's very difficult to think of every possible column that every researcher is going to want to have, right? So we try to encourage them, hey, you've got a different grouping of your samples. Could you put that in the batch column, right? Or you do this every time. How about a time column, all right? So try to use the columns you already have so we don't have a million columns. But right now, we're still ending up the text file so that they can still add in a few more things that are very experiment specific. part of this then was also the cell line curation tool, because cell lines are a mess. You know, is there a space, or is there no space, or is there a dash, or is there a semicolon, or is it a colon, you know, whatever. So went through and built a database of over 4,000 distinct cell lines. We also standardized the naming of custom lines. So we have the preferred sample name, and then, you know, someone, now it's a GFP variant, and it's a this gene mutant. You know, so the customized lines within the company, try to have a standardized way of naming those, both for other assays and for this as well as the parent-child mapping, which is helpful. Part of this then, we also have you know, integrated this with CCLE, Cellosaurus, and so we have the land names. So if we have data in a cell line and we want to map it to CCLE, we know what is it called in CCLE, because, you know, in CCLE they call it by what, disease cell line something. It's not just the name in the lands. And of course we standardize tissue disease names of the source of the cancer cell lines. And, you know, then we have just an admin mode, so we can go and fix things and adjust and add. But then the general users just look up, what do I call this? You know, H1975, what is that? Well, it's NCI-H1975. So they can move on, and we can have this database building of all the information coming together. Another thing is the problem of sample registration. Now, we just learned yesterday the magnificent tool from Joe of how you can take a design table and propagate that through an analysis to all the other counts. We didn't have that, and certainly that's tedious. So it was important for us to actually have the metadata with the NGS data initially, so that it came through everywhere. But then the problem with this, how do you register samples? Because it's one line per sample, but we may have, you know, 18 FASTQ files for that sample. So you need to have that organized, grouped, and concatenated into one cell. So what we had some of the group Ben Ellingson make was this grouper tool, which basically you just load up your sample file where you have all your FASTQ files and then the associated ID. Load that up. You can decide whether it's paired or not, because they have to be grouped differently. And then you'll get a converted file where it's concatenated those. So then you can just paste that column of file path into the sample registration. So as a result, now we're taking all the RNA-seq and all the NGS data and registering that as well for the future. So in terms of some different kinds of data we're bringing in, we're getting a lot of proteomics data now. And so what we've done is we're taking the MaxQuant data, we just load that as omic data, and we have as the ID column, first column, we have that as a UniProt ID. And then having built a custom UniProt annotation, server annotation, they can just load the omic data, you know, do the annotation, so now they know where it is and what chromosome and, you know, what gene ontology or whatever. And then go ahead and just do a simple QC. They can see if their samples are any good with the PCA, run a lemma, and then quickly, you know, use the standard tools, the volcano plots, the inference tests, Venn diagrams, gene set analysis, which is something they couldn't do before at all. They were just stuck in Excel and, you know, looking on the web. So that's been very helpful to the team. And phosphoproteomics, in this case, we're still doing this in our studio and loading the data in, but that gets to a question that Brett had asked earlier of how, as we go forward, how are we going to keep track of proteins and protein variants and different mappings, and also then post-translational modifications. How would we do that going forward in a unique way? Because you'd like that to be searchable, right? So in the same way, we can do a list analysis for every other kind of inference plot that we've generated. Something we've done quite a lot with is the L1000. So I forgotten whom showed the links database is here of the L1000, 1,000 genes with hundreds of compounds, hundreds of cell lines with all that data in a land. That platform is very convenient because whether or not you believe those 1,000 genes are perfectly representing all gene expression, it's still 1,000 genes, and it's a pretty good set. And so it's a 3D4 well plate. So you can just take a supernatant from a 3D4 well plate that's been treated and get your 1,000 genes per well. That affords the ability to do dose response for compounds across cell lines or multiple treatments with replication. So you can do that and then just use standard tools in Array Studio and then very quickly profile all your compounds in dose response with replicates and see, oh, hey, here's a section here where there's a gene signature that seems dose responsive that maybe is related to the on-target signature of that compound. And frequently then you can actually do the plots and see that you can get gene expression dose response that matches your other phenotypic response and really see the signature. And sometimes you can actually see, some compounds it's very pure. Some compounds that are off target, you'll see the dose response of your on target, and then you'll also see somewhere in the range some off target thing kick up, as you've gone into the off target and it's toxic above that level. So that's been very helpful. A variety of teams have used that to understand mechanism of action or look at the uniqueness of various compounds in a series for a particular target. CRISPR data, we've started that. Initially, we were just taking the counts from some external alignment and made a custom server annotation so that we could just bring in the CRISPR Library ID as the annotation, and then know what the gene was and where it was on the chromosome, because many times you'll get a knockdown from only a few of the reagents for that gene, not everywhere across the gene for whatever reason. And VOOM has been adequate to show which of the genes that are enriched or dropped out and in some kind of positive selection screen. So we're not using any fancy magic analysis or whatever. We just took the counts and do VOOM, and we can see really what's going on. And then we can use all the other tools once again, gene set analysis, whatever, to understand what's maybe driving those hits. And now, with Joe's help, and someone else, anyway, the support staff helped us. We worked out so we can use the single cell preprocessing to better do the adapter stripping and strip the barcodes. And so now we can just go from FASTQ all the way up through gene set analysis within the same tool, and then it's all searchable. Because certainly you'd like to say have some compounds that give you an inference test in some RNA-seq experiment, and then later on say, hey, those genes were in the same hit series from some CRISPR screen, right? That's the whole point of bringing everything together in an Omics Hub. As for clinical lands, we've worked with Sentient to do the whole exome sequencing, combine that with RNA-seq and the clinical parameters to make our own clinical lands. It was really nice to just both explore the relationships between the clinical parameters, between the clinical parameters and between the clinical and the omic data. As we've seen earlier, we've loaded some GWAS data at the summary level into genetics land. We hope to start using that more. It looks like it's really filling out nicely. And in this case, we've instituted take advantage of the controls. We have multiple levels of access to separate both the therapeutic areas, so one therapeutic area is going to look at its data and not the other, but also to separate those in translational biology that are actually generating the data or collecting the clinical data and doing the analysis, as well as those that want to just, or have the access to just view the lands and help make decisions about those clinical trials. So we were able to keep that all separate and keep everyone happy about who has access to what restricted data. We have that all set. The tools are here. It's just a matter of keeping track of who they are and then having it on the list. So if we could get the LDAP thing to move forward, I think then that'd be even better. Yes, so this is the also we're access controlled at Amazon as well. So it's secure. Everyone's happy with that as well, using the identity access management tools. So far, we only have 7 clinical trials, but we have made them all ourselves. So maybe we should have taken advantage of Matt's offers earlier. But so far, and it's certainly thousands of subjects. So that's really working out really well. Something else that we're using now is what I'm calling pharmacology genomics. That is, we have compound potency data for a large number of compounds across 160 cell lines. That's one of the sample measurement data that you can load against a land. So CCLE, for example, we can load these measurements against those cell lines. And because of that, now there's type ahead lookup of compound potency. So you're in CCLE land, rather than typing in a gene and seeing what the gene expression is, you can type in your favorite compound and see the potency of that compound across those cell lines. And you can use the analytics tools to search for genes with correlations between the compound potency, or between the compound potency and mutations or copy number. And so then you can plot, in this case, potency versus gene expression, and very quickly say, yeah, this might be the target, right? Or an anti-target, a resistance factor, right? So that this is really powerful and early discovery of seeing either what might be the target of some compound that we found in a phenotypic screen, or what's some standard of care compound. There's some standard of care compound. What's a resistance factor? So we're taking advantage of that as well, bringing in the pharmacology data. And well, yeah, as I just said, you can find resistance factors, markers, and the like. Now, one of the things that we saw earlier was these new web views of the lands. We tried to develop that for our own. We certainly have web services within the company, certain views where you can look up something about a project you'd like to see. Also, then for that target, where is it expressed? Is it differentially expressed? Any diseases? So in this case, we've got light web views based on human disease land. For the differential expression. So differential expression explorer, there's type ahead and intuitive selection for what's your diseases, and also what's your comparison type, disease versus normal. And then you'll have a bubble plot like disease land, but a little more streamlined, a little simpler. It's on the web. And then, of course, there are other really nice features. You can select the samples. There's the data table below. You can link out to the geo study if you'd like. There's some tool tips to identify, well, do I really like that study? And you may know that in this case, we're dealing with biologists, chemists. But then for this work to be accepted, the statisticians have to believe it. They want to know, is this study any good? That's typically the thing. So in this case, Brett Engelman made this, brought in study QC plots. So you can just right-click on that and say, all right, well, is this study very good? How's my p-value distribution? How many samples do we have in this? What's the separation in a PCA plot of of the treated control or the disease normal. So that's the flexibility that we have just in the light tool, sitting on top of on the land dumps. So that's the sort of thing that we're trying to pursue for the other data, the GTEx data, the TCGA, but obviously we're very interested in this web views that we saw as well. But nonetheless, it's a very helpful way in. And of course, we have here, if they just want to see, hey, there's this gene, is this differentially expressed in any diseases, is this important, whatever, they can see that. If they really want to get into it, there's a button. We can just go right into that exact view in disease land, because we have the URLs from OmicSoft that you can modify based on whatever gene and whatever view setup you want. So we have a URL that would take you directly to the normal disease land view for that. Part of this then is also getting this out there to the users is user support. So we're using Screen Steps, which is a program for making guides. Sometimes the guides are as simple as, oh, you want to do this, and it's a link out to a PDF from OmicSoft. Sometimes, the PDFs will have all the possible things you can do, but in this case, we just have a narrow, skipping some of the steps. You want to do this one and this one and this one. In the future, I think we'll start to use some workflows. Also something I picked up, yesterday, which is a very nice thing to be able to have workflows and share them. In this case, we're using Screen Steps to augment the tutorials in the wiki, and we can share them, we can link to them. It's a similar format to the PDFs with images from the menu, arrows to say do this and then that. And each one of these has a distinct URL, so we can embed them somewhere, or someone has a question, and we just send it to them. How do I install Ray Studio? Well, here, just this tells you how, or how do I who to contact, or whatever. So we've done quite a bit of that, and as well as some videos. So now I guess I'm just going to go over the current status or growth. So so far we've registered and processed almost 27,000 samples of internal preclinical and clinical projects. We've got 14 internal lands and 7 clinical lands that we've put together. So we're certainly familiar with the intricacies of getting a land curated, the data curated and loaded and properly converted. And we've managed and processed and integrated omic data of a variety of types: microarray, RNA-seq, DNA-seq, protein data, nanostring, as well as the EC50 data and the clinical outcomes. So we've been able to put all that together in one place. As I said, we started this in 2015, really. So at this point, this is 55 terabytes just of new data that's been transferred from outside vendors to our cloud. So just really amping up data from outside, migrating it in, and that's what's really great about the cloud storage. And at this point, we've got over 200 terabytes of total storage. So that includes backups, also results. That's the one thing that's interesting about NGS versus microarray data. As you process, the data doesn't get smaller. It just keeps getting the same size or bigger. So you go FASTQ to BAM to VCF. It's not getting any smaller, really. And so at this point, we're at 200 terabytes. And this is the kind of situation where being in the cloud has allowed us to do things. We have collaborators that say, oh, hey, there's this new collaboration. It's 2 terabytes. I can tell you in a big company, it's not that easy to say, hey, can I get an extra 2 terabytes of server storage? Like, you can't just do that in a day. Whereas with the cloud, you can just spin that up. And the other thing is that the number of active users is spanning, the number of active users has doubled every year for the last 2 years. So we started here in 2015, and we've doubled every year for the last 2 years. Right now, we're at about 150 active users in the second quarter of this year. And that's active users, not total users, because a lot of times researchers will spend some time researching a new target, and then they go away to the lab for 6 months, right? And then they don't come back. So that they come and go. but we're definitely expanding across the various sites and the various therapeutic areas. I had to drop the legend, but you can see that we have different groups: clinical data, non-clinical data. So we're still on a steep growth curve here. The team that's doing this here, so it's we're responsible for genomics and translational informatics. So it's Ben Ellingson who's doing a lot of the back-end work. Sadia is here. You've probably met her. Brad Engelman has done a lot of the work on the LANs and the web services. Aaron and Mark have been in the group for a while as well. So I guess my point here is with a fairly small team, we can do quite a lot in bringing all of these things together, and I think that's it's a testament to the product and all the effort from the OmicSoft team helping us get this done. And any questions at all about this sort of thing? Yes?
If you were starting again today,
Uh-huh.
would you take the same approach of building the land yourselves, or would you have taken up their offer to have them do it for
you? I think I'll speak for myself at least, I'm more of the type of person wanting to figure out how to do it myself. I think it would all depend on how much it cost, Certainly if it were free, no question. We have actually taken advantage of the services. We certainly had an external site with a custom chip, a custom Affymetrix chip that had a really excellent set of tissues for a human body map, but it was just a crazy chip, and it was annotated, I don't know, 05 something. So we took advantage of that to realign the probe sets, reannotate the chip, bring that in. So we have used the services for some of the land work, and I think it's likely we'd want to do that more. Certainly, I think it helps to do a little land work yourself, because sometimes the organizations need to spend a little time getting organized as to what's critical, what are our fields, what we want to bring in. I mean, you could spend time doing all that back and forth with the curation team, right? But it's probably more productive to kind of do a little of that yourselves beforehand, and then work with them, I think. I think I would have pushed hard to have our vocabularies be the exact same vocabularies as what OmicSoft is using. But if you pick one, you can always just map. That makes sense. Any other questions? Yeah.
So I think I didn't realize we actually have so many users, and I think based on our support emails, that it's actually not so many. How actually you deal with all your users? They have issues, actually solve that.
Well, so one of the things is that we early on started out really encouraging the users to CC us, or initially it was just me, when they contact you. Because frankly, when we started this, I didn't know NGS. I left gene expression before NGS was a thing. And I came back, and that was everything. How do we learn how to do these things? Or maybe it's just a simple thing. And so by having us CC'd on that, a common problem we've seen before, then we put that into our help file. And so rather than them contact your support, and then there's some back and forth about what's the problem and whatever. In this case, because we're directing them to us, we say, oh yeah, here's the link to the help file for that, or your link to the help file. If it's something we can't solve, then we will CC them and work with you to find the solution. And that's how we've handled that. So we've learned and built up, because sometimes it's just faster, because we just text them right back, do this. And they're like, oh yeah, right. So it's better for them getting it done. So that's maybe why our footprint seems smaller than it could be, if that makes sense.
Great. Very appreciated.
Well, I more have a comment instead of a question. I think this, even with the web portal that we are going to release, I think those little things, I won't say little things, those things that you build based on our tools, like the FastQ concatenation tool, the sample registration tools, those are, from my point of view, those are the things that are essentially the best complement to our tools, because it really helps the end users to be able to do things that we really don't do very well in practice. And even with the web portal, I can say that the existing web interface that you guys built, it will only be more useful than before, because you can have this kind of like in-place, internet-based view, but then right now you have a button called View in RStudio. You can also have another button say View in web portal, and then just link to like a web interface if they want to see more detail. Then we can have different layers that's designed for different level of like users.
Right. Yeah, and because that is one of the things that we've discovered is, I don't know if many of you may have used Spotfire. There's a lot of researchers, the first time they see Spotfire, they're like, oh, they can't deal with it. And eventually they love it, many of them, but some not. And it seems like RStudio is another level more complicated than that. So even people that are comfortable with Spotfire really are kind of put off a little bit by RStudio initially. And so the approach we've taken is to have a light view and then the full client, or in this case the web version of practically the full client, because some users just want to know, is this gene differentially expressed in any disease, or where, what tissue, and that's it. They're not going any deeper. Others, very quickly, then they want to get into the details, and then they want everything. Once they start to ask, so that's why we took that two-level, not a middle layer, but we're still open for how to do that. So maybe that web views will be a satisfactory, detailed view, or maybe it'll be a middle layer. Yeah.
I think for yours, you continue to use for
For casual users.
You are still using our data down, right?
Right.
Later you can use our RESTful API to query data real time. And also, then also, I think one thing we have in our mind that we want to have some guided query. Actually, some guided.
This is why you guys have a media,
right?
Right.
Something you know your users, you feel your guided.
Yeah. Right. And so if we can actually, with those help files, guided help files, have those workflows, which I've never used before. We used to tell them to turn that part off. That would be that'll be really helpful, because then we'd say, oh, well, here's the workflow for that. So they remember, click this, whatever. It's just as a further encouragement. They have that in the GUI as well as a help file to the side. That would work. And certainly these light web views are great, because we have web pages where someone comes up and they want to know, hey, here's a target. Do we have any compounds for that? Do we have any proteins for that? What's the project information around that target? They might also want to see, hey, where's that TCGA? And it'd be nice to have the same data that they see elsewhere, and that's what we have here with the Differential Expression Explorer. So, okay, well, any other questions? Nope. All right.